Thank you so much, Peter. That was far too kind an introduction. And I'm really happy to be here and will come and visit you anytime to see all the great things that you've built here at Queen's. So thank you guys for inviting me. It's lovely to see you all today. I'm going to be speaking to you today about one of the three programs that I run within the Singh Lab at McMaster University. So we focus on treatment refractory aggressive forms of brain cancer. And so we study treatment refractory adult glioblastoma. We study pediatric medulloblastoma, most often the recurrent MYC amplified group 3 type. And we also study brain metastasis. So, given that there's a lot of synergies with what people study here with my brain metastasis program, I really wanted to kind of showcase the work done by this incredible team. They're the smallest team in the lab, but I tell them that they're the high risk, high reward team, and they're small but mighty, and they've accomplished a huge amount. And so, I'm really happy to share their work with you today for this wonderful Nathan Kaufman visiting lectureship. So, oh, let's make sure I can move here. There we go. So I think I'll just sort of start by going over the fact that you can see here that deaths from lung cancer we know are the most frequent overall from cancer. But the fact is that these patients are not all dying of their primary lung cancer burden. In fact, these cancers are known notoriously for spreading and metastasizing, and 90% of these lung cancer related deaths can be attributed to metastasis. And lung cancer spread to the liver and to the bone and the brain. And once they've spread to the brain, survival is really terribly poor. We're looking at a 4 to 10 month overall survivorship, often with the only real treatment option, which often tends to be palliative radiotherapy. And basically, we know that brain metastases are the most common form of brain cancer. In fact, they're 10 times more common than primary brain cancers. And this rate is actually increasing because as we're going on to make these monumental cures of certain classes, subclasses of breast cancer and lung cancer and melanoma, and new agents we're developing are successfully holding the primary cancers at bay, these patients are actually going on to get brain metastases. And we think this is because as those cells that are seeding the primary cancer are probably traveling through that metastatic cascade and seeding the brain continuously. And once they get to the brain, it's a different microenvironment. They have different protections in that area, and they're able to survive the therapies. They've acquired new mutations that enable them to survive the therapies that are successful at eradicating the primary cancer, yet leaving these cells, sparing these cells to go on and form brain metastases. And you can see here that, of course, lung cancer is the most common cause of brain metastases, followed closely by breast cancer and melanoma, which is now almost tied with renal cell carcinoma. And then in fifth place, we also see colon to brain metastases. And although lung cancers make up the majority of brain metastases, we know that the standard of care for all of the brain metastases is actually quite lacking. The standard of care for a solitary large brain metastasis is surgical resection. And then after that is followed by radiotherapy or stereotactic radiosurgery for smaller brain mets and oligometastatic disease, as depicted on this MRI here. But in general, we have a great dearth of targeted therapies and therapies that could actually target the mutations that are driving these cancers. And so brain metastasis patients have a really dismal outcome right now. Again, that 4 to 10 months overall survivorship. And this really suggests that new therapies are desperately needed. And in order to be able to understand and develop new therapies for brain metastasis, we have to develop a greater understanding of the molecular machinery that's driving the spread of these cancers to the brain. And this is directly in line with the findings of a recent NCI workshop on brain metastasis that was published this past fall, really highlights the need for biomarkers to be able to identify patients in the first place who are even at risk of getting brain metastases. So really, we don't want to treat the cancer when it's already in its full-blown state. By the time your brain is seeded with metastases, there's more than you can even imagine that are apparent on the MRI. And really, that's treating at a point that's too late. What we really want to think about is the fact that those cells are beginning to spread from even the primary cancer's declaration. And so can we start thinking about treatment models that are not reactive, but actually more proactive and perhaps preventative or anticipatory or even interceptional? And so I'm going to share a few stories with you today from my brain metastasis program. But I want to highlight this fantastic paper that was published recently, the first author work of two amazing research associates in the lab, Dr. Sean Chafe and Dr. Kui Zhai. They were really incredible in driving this work, which actually took over 6 years in Singh Lab to set up and optimize this incredible in vivo CRISPR activation screen, to sort of develop the methods for this and to run it with our patient-derived human models. All of this was like building from the ground up. And Sean and Kui did an amazing job, carrying through to shedding more mechanistic insight than I ever thought we would have. So I'm just going to tell you a little bit more about their paper today. So to identify drivers of lung to brain metastasis, we first embarked on this in vivo CRISPR activation screen. So we made use of a clinically annotated human patient-derived lung adenocarcinoma line from my friend Charlie Swanton that had clinical annotation from the TRACER-X study. And we utilized the Calabrese library from the Dench Lab at the Broad Institute, which consists of about 58,000 guide RNAs targeting 19,000 genes at 3 guide RNAs per gene. And we use a library of 500x coverage. So we transduced cells with the library and then we inoculated them directly into the lung of mice using a modified thoracotomy procedure. So the tumor burden was then tracked over time by IVUS and the lungs and brains were removed at endpoint. And then we could really confirm, take the genomic DNA out and sequence it and determine the guide abundance and identity. So before I move to the next slide, I want to tell you just a tiny bit about the models that we've developed. So we are very lucky to, and during this talk, I'll try to shed some light on why I even started a brain metastasis program at McMaster in the first place. So when I first started the lab, we got over, this was back in 2007, we got over 100 brain tumor samples from our very clinically active, hardworking neurosurgeons, both on the adult side and the pediatric side. And, those samples were actually only representing the malignant tumors, as I had requested, please send me the aggressive ones from which we can form patient-derived lines and models. And so what I realized on evaluating those first few years of Singh Lab is that 50% of the tumors were from brain metastasis patients. And at that time, I was only planning to open glioblastoma and medulloblastoma programs in my lab. I thought 2 programs, perfect, 1 pediatric, 1 adult, that'll cover it. And then I realized that I almost felt this moral obligation to study this cancer that was so prevalent, for which we had no treatments, because the neurosurgeons were constantly resecting these large solitary mets out of these patients who otherwise would have died rapidly and bought them a few more months of time just to spend time with their families to get palliative radiotherapy and then to succumb to their disease. And I thought, this is terrible. We have to change the trajectory of this outcome. We have to study this. So that's how we started studying things. So we would get the patient-derived brain metastasis, bring it to Singh Lab. We would dissociate it enzymatically, recover cells that we would then grow in serum-free media and enrich for the stem-like population within those brain metastases. Then we derive new patient-derived xenograft models. And in these models, we would use orthotopic site injection of wherever that primary tumor was determined to come from. We'd grow them out for a week, then find out the pathology report, and we'd start to discern, is this a lung to brain, a breast to brain, or a melanoma to brain? And so then we would implant those cells back into their primary orthotopic site. So we would do an intrathoracic injection for lung. We would do a mammary fat pad injection for breast. We would do a subcutaneous injection for melanoma. And we would do intracranial as well, just to monitor and see what happens with intracranial growth. But the key thing to tell you is that these cells are already enriched for brain metastatic initiating capacity. If you were just to inject a cell line from ACGT, you would not get any seeding of the brain because the frequency of those metastatic initiating cells is very low in a commercially available cell line. But these are already enriched, taken from patients who have brain metastases, and therefore they're more likely to seed the brain of these animals. So we characterize these models, we develop them, and now we have them in the lab so that we can reliably and reproducibly get evidence of brain metastases, human brain metastases across these models. So a ton of work went into developing those models in the first 5 years of Singh Lab before anybody could even begin this project. So moving on now with the CRISPR screen, we started with 18,885 genes that were present in all of our cell inocula to be engrafted into mice. And then following engraftment, we were down to about 9,000 genes, give or take a few in the lungs at endpoint. And then when we actually assessed the guide RNA levels in the brain, we got ourselves down to about 400 guides targeting 400 genes. So then to shortlist the candidate driver genes, we filtered the hits detected in the brain with only a single guide, and that brought us down to 28 genes. So now here we are in the blue bucket. We then assessed clinical relevance using the TCGA and assessed by DepMap to identify genes whose loss actually impacted viability across a number of cancer types. And in the interest of time, I won't be able to go through the whole short list of our top 10 or 12 genes, but I want to share a little bit about the most exciting hit we found. And that hit is BASE1. So we identified a guide activating BASE1 expression, and this guide was enriched tenfold in the brains of the mice. So what is BASE1? You guys probably already know that BASE1 is beta-site amyloid precursor protein cleaving enzyme, which is a crucial enzyme primarily known as the first step in creating amyloid beta peptides, which form plaques in Alzheimer's disease. So this makes it a really major drug target. And it's an aspartyl and so we do know that it's a transmembrane protein that's mostly found in neurons, but also other cells. There are roles beyond Alzheimer's disease, and it's present in other neurodegenerative diseases. And there's also links to metabolic diseases like diabetes. So this has been the subject of a very intense clinical campaign with a number of pharmaceutical companies already involved in inhibitor development. And the thing I like about this is these are drug companies developing a drug for a clinical indication that is in the brain, so they're already blood-brain barrier penetrant. So that's great. So we know this, but we really know little about the role of BASE1 in cancer and even less about its role in brain metastasis, which really excited us about this target. So to assess the clinical relevance of BASE1 in non-small cell lung cancer, we stained a lung adenocarcinoma tissue microarray consisting of 69 tumors for BASE1. And you can see that BASE1 expression shows here in green, and it's highly expressed in roughly 2/3 of the tumors. And we next assessed whether BASE1 might be expressed in brain metastases arising from patients with lung adenocarcinoma. So we stained up 3 lung adenocarcinoma patient brain metastases, and BASE1, again shown in green, was highly expressed in these interrogated samples. And when you look at survival in these patients, from the TCGA based on BASE1 expression, you see patients with high levels of BASE1 expression in their tumors have decreased survival times. So BASE1 is clearly important for maintaining cell growth and self-renewal capacity of lung adenocarcinoma brain mets in vitro. We wanted to now assess whether targeting BASE1 would block the growth of these brain mets in vivo. So here we just took the BASE1 knockout cells and implanted them intracranially into mice, and we assessed tumor growth by bioluminescence. Oh, sorry. And you can see that, in fact, from these images, loss of BASE1 really significantly reduces tumor burden, and this is translated to a really good extension of survival. But now we want to assess whether we could impact metastatic brain tumor growth by targeting BASE activity pharmacologically. So now we're using the drug MK-8931, which is one of those BASE1 inhibitors from the previous slide. It's a blood-brain barrier penetrant drug that was trialed in Alzheimer's disease in patients. And so we inoculated the mice with cells into the brain. We let the tumors form for 7 days, after which time we gave daily oral administration of the drug. So administration of MK-8931 significantly reduced brain tumor burden, and this also led to an extension in survival. So our pharmacologic inhibition matches the genetic knockout. While these results are very encouraging, and this suggests we can target those established brain metastases with BASE1 inhibition, I did tell you earlier that I don't want to treat established brain mets. We want to see if we can actually intercept. And so we wanted to address whether we could even block the spread of lung adenocarcinoma to the brain. So to do this, now we made use of one of the other orthotopic models I told you about, the intrathoracic injection. We implanted the patient-derived CRISPR-activated BASE1 cells into the lung, and we allowed the tumors to form for 7 days, and then we began daily oral administration of MK-8931. Very importantly, we saw increased expression of BASE1, increased tumor burden in the brain, and MK-8931 significantly reduced this, suggesting that BASE1 targeting can be utilized to limit the spread of the primary lung cancer to the brain. To develop an appreciation for the biochemical changes that we think are instigated by BASE1 expression, we wanted to look at changes in cellular kinase activation in our BASE1 knockout lysates. So we identified significant decreases, you can see here, in ERK1 and 2 expression, and also in downstream c-Jin phosphorylation. And we confirmed these findings of the antibody array directly by Western blot in base 1 knockout cells. And consistent with the array, we saw that knockout of base 1 with 2 independent guides led to reduced MEK, ERK, and c-Jin activation, which was also observed when base 1 activity was inhibited by the drug. So based on our biochemical activity of base 1 as a protease, we hypothesized the contribution of base 1 to EGFR activation may actually be through cleavage. And as you guys know, EGFR is a very important driver of non-small cell lung cancer. And to test for this, we wanted to ascertain whether the two proteins localize near one another in a cell for this even to be possible. So to do this, of course, you guys know we used proximity ligation assays, which is, as you all know, is a technique that is capable of identifying when two proteins are within 40 nanometers of each other. As you can see from these micrographs of HEK cells transfected with the indicated plasmids, and you can see what the plasmids are, either base 1 EGFR or base 1 plus EGFR in the last panel. And then we stained for base 1 and EGFR, and we only detect foci in the cells where both of them are expressed. And I'm not showing it here, but we also confirmed that this under endogenous conditions in our lung to brain metastasis models. So based on this finding, we collaborated with the protease and mass spec expert, Dr. Antoine Dufreux at the University of Calgary, and he helped us to address whether incubation of recombinant base 1 with EGFR resulted in the detection of any cleavage peptides using this technique called TAILS, terminal amino isotropic labeling of substrates. So shown in the schematic, you can see the extracellular domain of EGFR on the right, and the identified cleavage sites are indicated with the blue arrows here. So you see all the blue arrows. And there are several different cleavage sites identified in domain 1, which you can appreciate from the structure of EGFR here on the right, and that's very important for the binding of EGF. We then decided to explore this in HEK293 cells where both genes are not normally expressed. So expression of base 1 and EGFR could readily be detected following transfection, and we then probe for EGFR and lysates with an antibody specific to the N-terminal isotope between, well, as you can see, increasing base 1 expression led to decreased detection of EGFR at the N-terminus in the whole cell lysates over here, which coincided with increased detection of the N-terminal fragments in the conditioned media. And this suggested to us that base 1 was being, that base 1 was cleaving in a base 1, cleavage was happening in a base 1 dependent manner. Now we wanted to assess whether the influence of base 1 on EGFR activity was important for its ability to actually enhance non-small cell lung carcinoma brain metastasis formation. So now we used another route of injection we like to use in my patient-derived xenograft modeling, which is the intracardiac route. I call this the unbiased route because you can place cells into the atrium and technically they should home to wherever they should home and not be influenced by other circulatory patterns. And so we really wanted to look at these intracardially injected H1299 base 1 knockout or control cells. We put them into the left ventricles of non-skid mice and followed them longitudinally with bioluminescent imaging. Once the health status of control mice began to decline, we called the entire cohort, we imaged their brains ex vivo, and you can see base 1 expressing non-small cell lung cancer cells readily seed the brains, whereas the knockout cells do not. However, when we restore EGFR signaling through expression, oops, sorry, that was over here. When we restore EGFR signaling through expression of the constitutively active EGFR mutant, the L858R mutant in base 1 knockout cells, we can restore the ability of these cells to seed the brains again. And this indicates that EGFR activity downstream of base 1 is really important for promoting its role in brain metastasis. So here I've presented the first in vivo CRISPR activation screen to our knowledge that was done in models of patient-derived brain metastasis to identify drivers of lung to brain metastasis. We identified base 1. It's a novel, clinically relevant, and actionable target. And we think base 1 cleaves EGFR to fuel a molecular circuit driven by MEK and ERK signaling to sustain these metastatic phenotypes. And it is possible, we think, to intervene at an earlier stage of disease where these patients, for example, are now cured of their primary lung cancer. They're in remission and they've been put on osimertinib or some other, EGFR inhibitor. And the amazing thing about that drug is that it blocks the recurrence of the primary lung cancer, but it does not block the presence or the development of brain metastasis. So at this stage, we're kind of wondering, is there a drug that we could put patients on that would accomplish the latter. So I'll come to that point later in my talk. So that's just a summary of what we did here. So now, having shared that first story with you from Sean and Kui, I want to tell you a little bit about the work of Daniel Mobilio. So Daniel Mobilio is an extremely talented PhD student in the lab who had the benefit of joining the Brain Mets team as an undergrad student and becoming what I would call like a supercharged graduate student. By the time he'd started his PhD, he'd already got 2 years of work on the Brain Mets team under his belt and was really incredibly driven to answer certain questions that had not been answered by the rest of the team. So to share Daniel's sort of research drive, you can see here, this is a beautiful model that he made for me. This is a depiction of a tumor cell. And in this case, the tumor cell is metastasizing to the brain. And we know that in the metastatic cascade, tumor cells have to undergo massive stress as they metastasize through the body. So we think they undergo EMT transitions. They are obviously transcriptionally reprogramming themselves to accommodate new environments. And they have to undergo shear stress forces in the circulation and withstand those. And so you can see that these tumors will then invade and seed and colonize the brain. And now they have to survive in a brand new environment that has its own set of metabolic stresses. And so Daniel's real question that he's driven by is, how do brain metastases and normal tissues differ at the metabolic level? And what is it about the brain that forms a good metabolic home for these cancer cells? So to understand how brain metastases differ from normal cells at the metabolic level, we went ahead to collaborate with Dr. Daniel Wall, a really good friend and another clinician scientist, a neuro-oncologist who is very, very well versed in immunology and in cancer metabolism. And so Dr. Eric Peterson, a postdoc in the lab, was able to help us profile the metabolomes of brain metastases from lung cancer and from melanoma and breast cancer. And we profiled them against normal human astrocytes. And so from profiling the metabolomes of the brain metastases and the normal cells, we used pathway enrichment analyses, and we found multiple metabolic pathways that were enriched in the brain metastases versus the normal human astrocytes, such as pyrimidine metabolism. There were hits in purine metabolism, the pentose phosphate pathway, Warburg effect, lots of different hits that we found here, and we were very intrigued. But the most intriguing difference we found was an altered or a sort of dichotomous pyrimidine state between the brain mets and the normal cells, wherein the brain metastases have a highly abundant level of UTP and CTP compared to normal brain cells. The normal brain cells actually have higher abundances of uridine and cytidine. In order to validate this further, here you can see this is, of course, and I had to go back to my med school days to try to remember this because now Daniel having found these new metabolic hits, I suddenly had to realize that I'm not a cancer metabolism person and had to quickly become one. And so here you can see this is the pathway of de novo pyrimidine synthesis. And this is the making of a new pyrimidine species, which is the only way to actually make it other than taking them in or scavenging them from the environment. So every metabolite in red here in this graphic are metabolites that we could detect in our metabolomics pipeline. And so each metabolite, here you can see in the pathway, all of these metabolites within de novo pyrimidine synthesis are higher across all of the brain mets compared to normal brain cells. And so you can see at every level of this pathway, here's all of the different metabolites detected, and they're always higher in the brain mets. And then we wanted to confirm this metabolism at the protein level. So we probed the most targetable enzyme in this pathway, which happens to be DHODH. And at the protein level, we see high expression of DHODH across multiple patient-derived brain metastases, lines here from breast, melanoma, and lung cancer. Now, when you're looking at the normal brain cells here, over here on this side, you can see that DHODH is not expressed. And this excited us because it's potential there's a therapeutic window here. And potentially we could target in a cancer selective manner. So we took the DHODH inhibitor that we were already familiar with. You guys may recall that there was a paper that came out of my medulloblastoma program published 3 years ago with Will Gwynn as the primary author. And there we recognized that DHODH was a targetable and essential vulnerability in group 3 MYC amplified medulloblastoma. So fortunately, we had on hand some of the BARE inhibitor, and we took this DHODH inhibitor and put it onto normal cells. And there was no effect even at the highest concentration. So even up to 20 micromolar, you could see that actually those normal human astrocytes and neural stem cells are just coasting by. They're not affected at all. But brain metastases from breast, lung, and melanoma were very, very sensitive to DHODH inhibition. All the IC50s here are in the low nanomolar range. So we wanted to assess whether there is any effect on tumor growth in vivo. So we intracranially now transplanted DHODH knockout cells as well as control cells from a patient-derived model of lung cancer to brain metastases. And we found that genetically depleting DHODH really does extend mouse survival in vivo. And actually, we went on to now assess the same thing with the Bayer compound, and we saw the exact same thing, that there's a great extension of survivorship. So again, genetic and pharmacologic inhibition showing an extension in survivorship. So Daniel is a very observant scientist, and he really does pay a lot of attention to all of his findings. I think this is the mark of a great scientist. It really comes down to skills of observation and seeing things that would otherwise not be in the direct line of your hypothesis, but you're noticing the side observation. Very often it's the side observation that leads to the big discovery. So Daniel noticed that when we give brain metastatic cells the DHODH inhibitor, the cells seem to have this lytic phenotype. And so on the right side of the slide are what the brain metastasis cells look like after DHODH inhibition. And Daniel was really wondering, what's the mechanism that's driving this type of cell death, and what is this type of cell death? And so first, we wanted to check if DHODH is actually inducing the typical mediators of cell death, such as caspase-3 cleavage, which is canonically an apoptotic signal. And we did find that DHODH inhibitor induced the activation of caspase-3. However, if you remember, what we all know about apoptosis is that cells turn into apoptotic blebs. But here we see the opposite. We're seeing more of a lytic phenotype. So it was recently discovered that caspase-3 can cleave gasdermin E, and therefore the amino fragment of gasdermin E keeps binding to itself, which then makes a pore in the membrane, which lyses the cells. And so you can see that here. So here's the gasdermin forming the pores, and you can actually measure this in real time if you label gasdermin E at the N-terminus. So here we labeled the N-terminus of gasdermin E with RFP, and therefore, if this type of cell death is being induced by gasdermin E, then you're going to see these labels. So here you can see the red foci are an indication that this type of cell death is happening, which we're now realizing that this gasdermin E driven cell death is not apoptosis, but rather pyroptosis. And so here, we went on to collaborate with Dr. Ray Truan's lab because, of course, he runs our wonderful super high resolution microscopy facility. So he did confocal microscopy, and I want to show you a video now of the DSMO control cells where gasdermin E is cytoplasmic, meaning there's no foci forming. But with DHODH inhibition, we see something different. So here you can see the cytoplasmic DHODH, gasdermin. And then if I show you now the DHODH inhibited cells, it's quite striking because here you can see cytoplasmic expression of gasdermin, but here you can see those punctate foci. So to us, this is really this evidence that you have these like multiple foci, gasdermin E foci means that pyroptosis is really happening. Oh, lunch is arriving. So here, I just want to show you why studying cancer cell death is important. I'll just go on to here. And the reason is because the way cells die is actually important and instructive to how you're going to treat cell death. And cell death can be immunogenic. And then cell death can also be apoptotic. And these cells are dying a non-inflammatory death. But pyroptosis is really a very immunogenic form of cell death, where the cells are lysing, they're releasing their inner contents into the environment. They serve as damps. They're damage-associated molecular proteins now. They activate the immune system. So this has a whole other set of implications. So in order to see the immunomodulatory effects of DHODH knockout in vivo, we intracranially injected our mouse lung brain metastasis cells now into black 6 mice to analyze the immune compartment in this immunoproficient model. We wanted to see the tumors at a single cell level And we actually went on to study these tumors by using our MIBI. We have one of the only MIBI scopes, multiplexed ion beam imaging at McMaster University. So here in these images, the different colors mark different cells. So NK cells are light green, T cells are pink, macrophages are yellow, the tumor is light pink. So you can see here, this is the AAVS1 control, where most of the immune cells are actually on the outside of the tumor. However, when you look at what happens when we do a DHODH knockout, now the immune cells are all within the tumor core. And then if we go ahead and actually quantify this now, I'll show you the quantification. You can see that we found T cells, NK cells, macrophages, all of the other CD45 positive immune cells are all at a higher abundance in the DHODH knockout tumors versus the control tumors. And then I just want to share with you this last observation from Daniel's immunocompetent mouse study. You can see the MIBI-TOF imaging. You can actually now do cell neighborhood analysis, and it labels the different areas of the tumor as certain phenotypes. But the cluster we're most interested in here is C6. That's the tumor immune infiltrate, and it's seen in the cyan color, which is composed of T cells and macrophages and NK cells. And again, you can see here that the DHODH knockout tumors have a higher, much higher frequency of cluster 6. Again, this suggests that the DHODH knockout is immunomodulatory, and it stimulates these anti-tumor immune responses. So with all of this work now, Daniel is landing on some sort of key mechanistic studies to complete his work, and we're very, very excited about the implications of his amazing study. And I think he really did get some answers on what was different about the brain metabolism. So at this point, I want to sort of finish on a last story that I think is exciting because it's translational and really comes back to that concept I raised earlier about can we treat metastases in a more anticipatory or interceptional fashion, especially given the fact that we have now harnessed these models that can give us a little bit of an earlier insight into the very beginnings of brain metastasis formation. Before I do that, I'm going to tell you a little bit about therapeutic targeting head-on. And so the exciting thing about my lab with the 3 different programs is that there's a lot of crosstalk between the programs. And the nice thing is that there's a lot of sort of facilitation of when people sort of make an observation, oh, GBM team is noticing this. I wonder if it's going to hold true for brain metastases. There's a lot of really nice collaboration that happens between the teams. So I think you guys will remember several years ago now, we published a paper where we had developed an approach for glioblastoma targeting CD133, which was the antigen 5 transmembrane pentaspan protein that we first used to purify or enrich for brain tumor initiating cells. We recognized that this receptor could be a very good target to kill them as well. And we did this proof of concept study where we developed CAR Ts and bispecific T cell engagers, as well as using the naked immunoglobulin against CD133, a beautiful antibody called RW03, and we showed how effective that was in glioblastoma. Well, a very, very talented PhD student in the lab, Agata Kulizik, had her eye on this, and she began to wonder if CD133, which tends to correlate with very poor prognosis, with aggressive and invasive spread of cancers, and actually identifies cancer stem cells across 11 to 13 different cancers, she wondered if this could also be, CD133 was also expressed in brain metastases. So she went on to actually take the CD133 CAR T and test it in our brain metastases models, and very excitingly found that in fact it does work in some of our models. And here you can see her findings. This is the preclinical efficacy, and you can see that there's a 20% survivorship when we treat the lung to brain metastasis mice with CD133 CAR T. But even more exciting, look at the results with the colon to brain. So this does not surprise me, and I'll take you back to my days when I did my PhD in the lab of Peter Dirks, when I was first learning how to do flow cytometry and fluorescent activated cell sorting using the CD133 antibody to purify and enrich for those brain tumor initiating cells. And there was a very, very, very talented surgical resident named Catherine O'Brien, who was working in John Dick's lab at the time. And we were kind of doing our PhDs in parallel. And she came over to me and said, I'm just working on these colon cancers, because that's what I'm going to study for the rest of my life, colorectal cancers. And I'm just wondering, because you're dissociating solid tumors and flowing them and looking at marker expression. Do you think they express CD133? So I remember at the time I taught Catherine how to do FACS for CD133. And then she ended up publishing a Nature paper on the identification of colon cancer initiating cells, which did express CD133. So I told Catherine about these findings, and she was not surprised at all that colon to brain metastases should be well targeted by our CD133 CAR T cell. So it's nice when something from 20 years ago actually comes to bear. So another very sort of exciting project that I just wanted to mention regarding CAR Ts is that we've also collaborated with our wonderful colleague, Thomas Kisslinger. So the best way to figure out if your CAR T cell is going to target something is not to do transcriptional level profiling, and please don't do single cell, it's not helpful. The most informative thing is proteomics, because you want to know if your protein, in fact, the most, even better is glycoproteomics. So if you know that something is expressed at the cell surface, 80% of the hits from Thomas's glycoproteome platforms actually are expressed and are targetable by CAR Ts. And so we had already been collaborating with Thomas, and we helped them to demonstrate the therapeutic utility of their N-glycoproteomics platform. So this was a real tour de force study by Thomas's very talented postdoctoral fellow, Manusha. And Manusha basically profiled the cell surface of patient-derived xenografts across 7 different tumor types. They identified the cell surface expression of the protocadherin FAT2 as being upregulated in squamous cell carcinomas relative to normal tissues. And they actually developed an antibody together with Admari Innovations, and then Sean Chafe from my lab built them a CAR T cell using our 2nd generation backbone against FAT2. And we found that in addition to working really well against head and neck squamous cell carcinomas, of course, we had to trial it in our brain metastases models. So we found out that the FAT2 CAR T also worked well in lung to brain metastases. And again, here we use the same intracranial delivery model that I've already discussed. So this paper was is just in press, and it was just a really nice collaboration between Thomas's lab and our lab. So I'm going to finish on possibly the most exciting and translational program in our lab. And this is, of course, a program developed by during the PhD thesis of Agata Kulizic. So we know that every year 5 million people are diagnosed with a type of cancer that has a high risk of spreading to the brain. 30% of all cancer patients eventually do die of brain metastases. And we know that there's no effective therapeutic options. I mentioned earlier, there's no really good target therapies, no molecular therapies. And the bulk of these cases, of course, arise from lung, breast, and melanoma to the brain. So this program began probably over 15 years ago now, when the Singh lab first began to characterize this cancer cell population in the brain, that, as I've already alluded to, the brain metastatic initiating cells. These are the stem-like cells that we think actually drive the growth of these cells through the metastatic cascade. They're able to survive every stage and seed the brain. And what we did is we actually profiled these cells from all of our different models. We were able to capture the stage where mice were dying of their primary tumor burden, and we would harvest back their brains. The brains looked histologically, radiologically normal, but when we flow sorted the entire brain back, we would find 1 to 2% of cells expressing EpCAM, which is not normally something that's in the brain. And we realized these are cells seeding from the epithelial primary tumors. And so we were able to profile those cells, which are the first pioneers or invaders into the brain. And we found very excitingly that across all of the different cancers, there was a universal I'm homing to the brain signature that we found in what we define this now as the pre-metastatic state. So these are the cells that are capable of invading the brain, and they had a universal pan signature. So what did Agata do? I thought something very clever. She decided to use the Broad's connectivity map program to actually map against the signature to figure out what drugs would reverse this I'm homing to the brain and invading the brain signature. And from that, this universal profile, we were able to get a list of drugs that would theoretically block the spread of the primary cancers to the brain. And one of these drugs was actually mycophenolic acid. And you guys know that that's a drug that blocks some of the critical mediators of de novo purine or GTP synthesis. So we actually realized that the rate-limiting enzyme in this pathway is IMPDH. And potentially this is a potential therapeutic target for brain metastases. So we went on to confirm this using known IMPDH inhibitors against the different types of B-mix from lung, breast, and melanoma. And now we're developing drugs that block this pathway to stop the replication of the B-mix and prevent the formation of brain metastases. So in parallel and out of necessity, we're also developing biomarkers that could be used to identify patients who are in remission from their primary cancer, but at high risk for developing brain metastases. So IMPDH is a clinically validated therapeutic drug, and there are drugs on the market for immune suppression following organ transplantation. And in fact, you guys probably know mycophenolic acid is used as immunosuppressant in renal transplant patients. But some of these IMPDH inhibitors were previously evaluated in late-stage oncology trials, but they failed because of dose-limiting immunosuppression being a major drawback. So these previously developed inhibitors are also not CNS penetrant, and they were developed in, again, not as anti-cancer drugs, but this makes them kind of a poor choice for cancer patients. You don't want to immunosuppress further a cancer patient. So we are now developing selective and potent drugs. And by we, I mean Professor Jake McGolan, who's our fantastic medicinal chemist who's embedded within the Faculty of Health Sciences at McMaster University. He sat on Agata's committee, and he really, really helped us with this like fundamental challenge of developing CNS penetrant IMPDH inhibitors that may somehow be selective for blocking brain metastatic ability, but not activating immune suppression. So you guys may know there's two IMPDH isoenzymes, and it's recently established they have distinct expression profiles. So IMPDH1 is crucial in the immune cells, but IMPDH2 is actually essential for regulating brain metastasis initiating cell activity. So we think we can possibly segregate the activity defined by those two isoenzymes. And in our lab, we've actually found multiple lines of evidence confirming a key role for IMPDH2 specifically in driving brain metastasis. And this provides a very solid case for developing a new class of inhibitors and potentially being able to commercialize them. So we confirmed that really we do have very good platform potential because we're comparing them against known IMPDH inhibitors. And another exciting thing to tell you is that, remember from our in vivo genome-wide CRISPR activation screen, I told you I couldn't discuss all the other mediators, right? But the very exciting thing is that in that screen, we pinpointed one of the top hits was IMPDH2, but not IMPDH1, which was like number 800 on the list or something like that. So we actually had quite a bit of independent validation there. And we're trying to now generate very, very robust target validation data. We want to show that IMPDH2 knockout in B-mix is sufficient to block their growth both in vitro and in vivo. And we've been very lucky to win a CIHR commercialization grant for this program. We won the McMaster Seed funding, and by we, I mean Agata, I should say Agata. And she won the seed grant competition. And we have been able to spin out a company that is called Block Biosciences. And Block is currently developing this class of IMPDH inhibitors, as well as the biomarkers to define the patients who would be at risk and who would be potentially treated by these. And really, if you think about what our vision is, I mentioned earlier that class of lung cancer patients in remission who are on osimertinib, they're not going to die of lung cancer burden, but they're still going to get brain cancer, brain metastases. So what if we could have a pill available at diagnosis of the primary cancer? Lung cancer is most likely to migrate to the brain within 1 year. If a patient is going to get brain mets, they almost always declare themselves, as opposed to breast cancer, which is more indolent. You might wait 10 years to get a brain met from breast cancer. But in lung cancer, there's this 1-year window. So this would be our first indication, the non-small cell lung cancer patients. And can we stop the metastases to the brain with this drug strategy? So a biopsy of a lung tumor could actually identify a patient who's at high risk for brain metastases using our predictive biomarker analysis that we're developing. Then an oncologist could provide a safe plan for taking the pill based on this risk and the specifics of the lung cancer treatment. We've already done studies in the lab to show that the drugs that we're developing could be compatible and in fact even synergistic or additive with osimertinib. So now you're envisioning a treatment strategy that would be very similar to how you take a statin to prevent heart disease. Would you now be on this drug? And something the drug companies probably would like is you could be on this drug for life, simply to keep, proactively and preventatively blocking the spread of your cancer to the brain. So this initiative began with a research collaboration, again, between my lab and the lab of Dr. Jake McGolan, and we're the scientific founders of the company, but it's really Agata who's taking this home now, and she's now the full-time scientific director of Block Bio. And so through this very productive partnership that we had originally with Admari Bioinnovations, we had the great pleasure of working with Dr. Amy Finney, who is part of the Admari team. Amy's now left Admari, and she's become full-time CEO at Block Biosciences. we really benefit from both her scientific and her business knowledge. So our co-founders bring a lot of science and now business experience. And we've already been sort of seeking out a lot of key opinion leaders with clinical expertise in lung cancer to find out if they would endorse the strategy. So this is, you can check out our website online. We've just sort of got all of this going, which is super, super exciting. So on that note, I want to end and thank all of our amazing collaborators. First and foremost, the wonderful students whose work I showcased today, Daniel, Agatha, Sean, Kui. Again, small team, but very mighty. And the body of the work that they built is really impressive and hopefully going to change outcomes for patients in the future. And with that, I will thank everybody in Singh Lab on all of the different teams, and thank you for your attention.
Thank you very much, Sheila, and we have time for questions, so I'm sure there'll be lots. Andrew, go ahead.
Fantastic talk,
Thank
really
you,
exciting,
Andrew.
and really hopeful for your spin-off company
Thank you.
to be able to make that kind of progress in a very important set of patients that we want to have live longer lives after lung cancer. Just wondering if the dormancy of metastatic kind of brain metastatic cells, if you think there's a kind of fundamental difference in the wiring of lung to brain versus, say, breast to brain and or colon to brain.
Absolutely. I mean,
is there a role for trying to understand dormancy as a problem in kind of seeding of the brain?
100%. So I love the fact that you use the word dormancy, because I think all the implications of the molecular mechanisms underpinning studies of dormancy need to be applied specifically to breast, which with its indolence of forming brain metastases and colon. I really do think so. And again, we were very focused just on one thing when we were analyzing all of our pan signatures, but we could easily go back and start to look more in depth for other indicators. Really, we were just looking very single-mindedly for what's the signal for driving it, for seeding the brain, right? But now we can potentially go back and interrogate those data sets for other important questions. Yeah. And there's a very incredible group of scientists at Harvard who are looking at, I don't know if you're familiar with them, they're a wonderful group, and they're very focused on breast to brain metastases. And so we're looking at exploring some of their patient cohorts and data as well once we mined our signatures.
Thank you for your talk. Could you comment a little bit on what is your impression of site-specific metastasis versus perhaps higher metastatic potential of the cancer cell in general? What makes me ask the question is that there is many cell lines that are grown, that are developed to be brain homing, liver homing, lung homing, and in my personal experience, when we grow them orthotopically, many of them just metastasize everywhere.
Yes, yes.
What is your impression of that? And then perhaps related, when you talk about brain metastases, do you think those are coming from primary tumors or, well, and lung tumors is perhaps a little bit different, or is it secondary metastasis from lung metastasis? Because, you know, I guess there is that dogma that lung capillary bed is what primary tumors of other organs see first. And I don't know, what is your general impression of?
Yeah, those are all excellent questions. I think those are all the key challenges in the field, actually, as you listed them off. So maybe I'll start with the first one. I think Joan Massaguet and all of the people he's trained have been developing now for some time these site-specific organ metastases signatures. And, you know, our friend Peter Siegel, you know, has started his own lab based entirely on the one program. And I think it's very smart to sort of focus. However, having said that, I think it's highly probable that those signatures are not specific to one organ only, and in fact probably change over time. And we haven't studied like the temporal evolution of these signatures either. When do you study the signatures, right? Like clearly the metastatic cascade, we think we're just studying different steps. But remember, that cascade starts the minute the primary tumor forms, and it's already probably shedding cells that are metastasizing everywhere. And I think there's even an immune component to that question. Why is it that our immune cells will kill some of those cells in circulation but spare others, right? So I think there's just a huge host of questions, which means that more of us have to be studying this. Because to be completely honest, the most common question I get from reviewers is, okay, so this is what the brain metastases are. What about all the other organ metastases? And then I have to clarify that I'm a neurosurgeon and I generally only care about the brain. But truly, there's many other outlying questions. And I think we need to form a bigger community to answer these questions, and more people need to study metastasis overall, right? So I would really love to see more metastasis grants at CIHR for that reason, because I can't answer all those questions.
Me?
Charles, yeah, go ahead.
Oh, thank you. I agree with Andrew. This is very exciting.
Thank you.
Thank you for coming here to give your talk. Very interesting. My question has to do with your xenograft model.
Yes.
The establishment of those cell lines, that metastasizing to the brain. Is there a concern that through the process of in vitro culture and then putting them into a mouse, you're selecting for cell lines, I guess, that metastasize using single mechanisms?
1,000%. Yeah.
Yeah, and whereas it's recognized that metastasis is probably a multifactorial, and that's why often, single drugs don't work, and because there is often development of resistance to them, because
Yes.
you can enrich or select for subpopulations of cells that will metastasize via alternative pathways.
Yeah.
It has to do with the model too, I guess, but also your model is immunodeficient. And so what are your thoughts on that?
Yeah, so
that you are not ignoring, I should say.
No, you're right. No, every model has flaws, and you listed exactly correctly all of the flaws of our model. So I think the only way you can offset that is to correlate it with other data sets that don't have those flaws. We're never going to be able to have a perfect model, right? And so in my experience with syngeneic models, or the models that are out there that are only mouse, very often you find those mouse hits don't extrapolate or translate to human biology, right? So you're kind of in every direction, there's a flaw in every model. So
That's not the challenge.
yeah.
So not criticism on your research.
Yeah, no, I totally agree. No, the reason I'm acting in dismay because this is like what you're identifying is a big problem for us, right? So what we're trying to do is correlate the hits that we get with actual patient-derived data. And there, what we run into is there's not a lot of human patient-derived brain metastases that are collected and sequenced or, out there for profiling. So Sean in the lab is actually trying to access from multiple different sources, and we have partners in Quebec and from different brain banks. We're trying to make a tissue microarray of patients who have lung to brain metastases, and then we can validate the hits in actual patient samples. And then I have a little more faith that something I'm seeing is not, a sort of confounding effect from a mouse model or a PDX model.
Potential issue with that, though, is not the fact that there may be cryptic subpopulations even within the brain metastasis that use alternative mechanisms, so like phenotyping the bulk of the cells, may be useful, but the problem is those subpopulations.
So the way to answer that question is twofold. One is something we've done in our GBM program, which is barcoding. So we have done barcoding, and what we recognize from all of the models, the in vivo models that we've set up, is, to your point earlier, only 10% of cells actually that are injected into the brain in a GBM model are capable of initiating tumors again. And part of that could just be because of the artifact that you're saying is that they just like to form tumors in a mouse brain, right? And might not even replicate in humans. But if you barcode these cells, then we could certainly observe which clones are capable and which subsets of the cells are capable of actually seeding the brain. So we could drop a barcode library at the very beginning of our metastatic cascade and then observe what clones arrive in the brain. So that's one thing. The second thing is my friend Charlie Swanton, and this answers your question as well, has published some beautiful clonal phylogeny papers where he's done mutational profiling and dissected all of the different clones. And what he found is, and it's very disarming and unsettling what he found, of course, is that, you have lung cancer metastasizes to all the different organs, and every different organ has different subpopulations of clones with different drug sensitivities, right? So he's done some really seminal work to document that. And I think documenting that kind of level of heterogeneity is critical for us to begin to imagine how we treat it. He didn't offer any solutions in his paper. He just said, whoa, look at this explosion of clones that are not treatable. But at least I think documenting and, looking it in the face is the first step.
Thank you.
Cool.
Thank you for your talk. I really enjoyed it. A couple questions, sort of unrelated. The first one being sort of similar to what Dr. Graham had to say, but for the base one inhibitors, you guys did a great job with the initial study, but is there a way to study if you kind of have sort of that clonal resistance over time in the mouse models? And then I guess another question for the glycoproteomics, is it the specific glycan that you're looking for, which is telling you that your targets are on the surface? Like, how are you determining that your proteomics are surface specific?
Yeah, so that's more of a question for Dr. Kisslinger. As far as I know, he has different tags and labels that all represent all of the different isomers of the glycoproteome. And so that's a really technical question I don't think I can answer, but I know he can. So I would probably direct that question to him. And for your first question, yes, it's a very good question. That's something we do have to monitor over time because all of drugs, tyrosine kinase inhibitors, awesome, they're all prone to developing very quick resistance, right? And these cells are going to escape mechanisms, even if we're treating with 2 drugs, there'll be escape mechanisms, and we're already kind of poised to figure out how to get ahead of that. So right now we're modeling and mapping what happens when we treat with both cells, both inhibitors, and then inject them into mice, and now what escape patterns happen. And so we're definitely going to have to continue profiling. And Sean has actually helped me to write another Cancer Research Society grant to follow up on the mechanism as you're suggesting. So still way more work to be done.
Go ahead, Michael.
Thank you. I was really interested in the colorectal specificity of some of the later working inhibitors. And I was wondering, just moving back to, I guess, the top of the stem cell hierarchy, like what makes the colon so similar to the brain? Like if I had to guess, I would assume the lung and the colon would be a lot more similar, like epithelial, adenocarcinoma. What makes the colon take on, or I guess what makes the brain maybe take on?
So I would say, I think in terms of the clonal hierarchy, it's way better fleshed out in colon cancer than it is in brain cancer. I still think our brain cancer hierarchy is very rudimentary. But if you look at the work of Hans Klevers, he's even gone to the point of like identifying the topographic like position of the cell in the crypt can determine its cell fate and identity. And so I think it's much more cleanly mapped in the colon. So I don't know so much if there's similarity between colon and brain, or whether it's just that CD133 is a very good marker for detecting these, invasive treatment refractory cells that can evade current chemo and radiotherapy. And I know Catherine has gone on to do subsequent very beautiful work at looking at these persister cells, right? The cells that evade chemo and radiation in colon cancer and go on to, seed treatment refractory disease. So to be honest, I think we could probably, still apply some of the paradigms from colon cancer to brain to learn more.
Okay.
Yeah, so first of all, great talk. I think your work is really fascinating.
Thank you.
So my first question is, when you give your CAR T cells, I noticed you give them intracranial.
Yes.
Do you know if this still works if you give it intravenously? And likewise, when you give it into the brain, do you see CAR T cells able to backtrack into the bloodstream through the blood-brain barrier?
Yes, they do. So the blood-brain barrier is much more permeable. So the brain is not, according to dogma, as immunoprotected as we think. And we know now that there are these dural lymphatics and other structures identified by groundbreaking colleagues that actually permit traffic of normal immune cells very well across the brain, blood-brain barrier, as well as CAR T cells. So the reason that I want to put them into the brain, and the reason we do it in our models, is because that's how I intend to put them into humans. So we're going to be implanting Ommaya reservoirs into patients to put the CAR T cells local regional with patients with brain tumors. And I really think it's the best way to treat brain cancer is to do it local regionally at the site. And that's now becoming the more trusted way of putting CAR T cells into brain cancer patients. And there's many, many trials now that are delivering that way. So is there bidirectional crossing? Yes. But some of what we can do is, depending on the target we're going after, putting it into the brain constrains most of the activities to the target in the brain, and you're actually going to get less off-tumor, on-target systemic toxicity now, because the CAR is not effluxing across the blood-brain barrier as much as if you had put it IV. So in some cases, you want to constrain the activity to the brain, get less systemic tox, then it works well. So having said that, we rarely do IV injections of CAR Ts. because we're kind of trying to gear our data towards supporting that approach in humans.
Follow-up, go ahead.
Just the second question that's kind of like on the same topic, but not exactly. That was when you do your previous CAR T cell work has been on immunodeficient, correct?
Yes.
So how do you predict that having an immunocompetent model might change that, especially given I know there's a role for microglia, especially in creating a
Yeah, that's a great question. So we have a paper that's just been resubmitted on Friday. I really hope it goes through and you can read it soon. But we've done some work in immunocompetent models with CAR T cells against a target called GPNMB. And also we've developed humanized mouse models where in our paper on CD133, we actually humanized the bone marrow of mice. And then we were able to observe if the CD133 CAR T had toxicity against the normal hematopoietic stem cell niche, for example. And so there are models that you can develop. Unfortunately, they're hideously expensive and very hard to work with in terms of getting enough experimental reproducibility with large cohorts of mice. But yeah, there's a lot of ways to improve those models and answer those questions.
I'm gonna ask the last question. So that's very fascinating about the base one cleavage of EGFR. I'm guessing that Dr. Dufour found other things that were differentially cleaved in that system. Are there going to be other stories? Because the EGFR story is fascinating,
Yeah.
especially because it sounds kind of counterintuitive where you're getting a reduction in ERK signaling.
Yes.
But the downstream effects of that can include things like dormancy or changes in expression of all kinds of players that could contribute to not just the metastasis, but to how those mets grow. And I was fascinated by your description of sorting brain and finding all of these cells. And I'm guessing that they represent a very significant proportion of those metastatic cells, but they're not necessarily a big part of the tumors that you see, the met
Exactly.
that you see.
Yeah.
So there's probably a lot of other information in there.
I totally agree. And just as I mentioned as an answer to one of the earlier questions, a lot of our data sets are not completely explored. Like we have to go back and pick up more information. And I think it'd be lovely for Sean to talk to you a little bit more about the work that he's proposing for this new CRS grant. But we've got Sarah Andres involved. We're looking at a structural biology perspective, conformational changes of VEGFR. Like there's a lot of follow-up to do to the work and to answer the questions you're asking. And I'm hoping actually that Sean may start a brain metastasis lab at some point and take a lot of these things with him. Because as I mentioned earlier, we need more people to study metastasis.
Well, I'm cognizant of the possibility you must be starving by now. So it's torture to have you probably smell your lunch behind you. So please join me in thanking Sheila for coming up and giving a wonderful talk and spending the day with us. And she's going to get a chance to meet with some of our trainees now over lunch. So
thank you for inviting me, Peter.